and welcome to the Drexel Interview. I'm your host, Paula Morantz-Cohen, speaking to you from Drexel University's Picture Gallery. Today our guest is Craig Venter, a pioneer in gene research and founder and president of the J. Craig Venter Institute in Washington, D.C. In 2001, Dr. Venter astonished the scientific world by publishing the completed sequence of the human genome. The repercussions of his work are enormous. Craig Venter, welcome to the Drexel Interview.
Thank you. It's nice to be with you.
Well, first of all, there's been so much publicity about the genome and the sequencing of the human genome, but I think some people, including myself, are not entirely clear about what a genome is and what it means to sequence the human genome. I wonder if for our viewers you could explain a little bit what that is.
Sure. Our genome is our complete collection of genetic material that we inherited from our parents. It's in the form of our 46 chromosomes inside of every cell. So we get half of our genetic code from our father and half from our mother. And there's roughly 6 billion letters, A, C's, G's, and T's in our genome. So decoding it was working out the exact order, the spelling of all 6 billion of those letters. That's why it was such a big task. There are a lot of
massive task. And could you explain a little bit what the antecedents to what you did were? What were the building blocks to your contribution? And what specifically you brought to this task?
Well, like all science, it builds on what became before. So we had new sequencing instruments that teams of engineers had made. We had to build very large computers, the third largest in the U.S. in 1999 to actually assemble the genetic code. But my team brought to a whole new approach where we used mathematics, robotics, and high-end computing to replace tens of thousands of scientists that people thought it would take because the task was so large. It had to be split up not only in the U.S. but around the world.
So I know in your book you refer to the shotgun approach.
That's right. We call it shotgun sequencing.
And this was looked upon askance by the by the purist side of the scientific world. Explain to me why it was, what it involves. I mean, I know we probably can't understand that well.
They just thought it was impossible to do because it's such a large calculation that we did in the computer. Yeah. So at the start of the Genome Project in the mid-'80s, early '90s, the best algorithms could only deal with 1,000 sequences. And to sequence the human genome, we did 27 million sequences. So you can see why some of the scientists reviewing our proposal thought it would be impossible to mathematically assemble that many.
And how did you know that it would be possible?
Well, we started in 1995. We sequenced the first genome in history using this technique, and it changed genome projects even for bacteria from 13 years down to 4 months. And it was obvious from that point on to me that these are the techniques we'd use for the human genome, and that's what happened 5 years later.
So even though you had used this approach on other forms of life, and that had been proven, and you had papers to prove it, there was still great skepticism in the scientific community about it.
Well, the scale was very different. So the first genome we did was only 1.8 million letters of genetic code. And they thought that would be impossible with 25,000 sequences. And we had to scale it up to 27 million sequences for human. So I think it was just beyond the imagination of the people that were doing their smaller projects that anything could happen in a small lab. And in the end, instead of requiring most major governments and $5 billion and tens of thousands of scientists, we had a small team of just a few hundred of us in one building that sequenced the human genome. We set up a large number of machines to do it, a very large computer. But that's the problem with science. It's introducing new ideas is usually very difficult in science. It's quite often met with extreme skepticism, but the onus is then on the people with the ideas to try and prove that their ideas are right. The problem is in these areas of research it takes lots of money and large teams to do that. So unless you have a unique opportunity, you don't ever get the chance.
Maybe you can tell us a little bit about the applications of what you have discovered. And right now, for example, there have been so many articles lately about new companies that will be able to sequence one's own genome. And you did this, you sequenced your own genome, actually, as part of this, this great innovation here.
Well, in fact, I was one of 5 initial people that it was a complex mixture of all 5 of us.
It was a composite,
so
yeah.
It was 3 women and 2 men, people of self-described ethnicity as African American, Hispanic, Chinese, and Caucasian. In fact, because we did this complex mixture, a lot of the variation got subtracted out 7 years ago. The public project only did half the human genome. It was called a haploid genome, thinking you could infer what the other half is. In fact, you can't from that. And so in September of this year, we published the first complete human genome, and that was my genome, and it had the complete set of chromosomes from both of my parents. And from that, we found out that we're far more different from each other than we thought 7 years ago. 7 years ago, we thought we all had the same sets of genes, and we differed only in 1 letter out of 1,000 in that genetic code. Now we know you and I are at least 1 to 2% different, which is a huge change. In fact, up until recently, we thought we were only 1.27% different from chimpanzees.
And what is the difference between us and chimpanzees?
So now that's changed too, fortunately. That's 5 to 6% difference.
Okay.
So we had trouble 7 years ago explaining all the variations in humanity based on these minor changes. It makes a lot more sense now, and we have now the true blueprint of a single person. And in a very short time, we're scaling it up to do many more. You referred to companies that are out there. All these new companies are just measuring part of the genetic changes. So out of the earlier work, we thought it was just these single nucleotide polymorphisms, the 1-letter changes, changing an A to a C, for example, or a G to a T. That would be a single nucleotide polymorphism. That's what companies were setting up to measure. But it only measures maybe 1/3 of the actual changes in the genome. So it's starting down the road,
but it's not there.
But it's a long way from it.
But there are things that we can learn now.
Well, we can learn some things about ourselves, you know, from my genetic code. I describe that in my book, and boxes throughout the book explaining some genes and some traits. And I use examples to show how complex it actually is. So the worry with the gene chips, the companies that are doing things now, incomplete information could actually be dangerous because it's only partial information. And what we find is if you have risk for disease in one set of genes, you may have another set that totally compensates for that and removes that risk or makes the risk worse. You know, we're at the earliest stages of trying to interpret the human genome.
So the notion that we can pay $1,000 and wait 2 weeks and get some reading on some aspect of ourselves is unlikely at this point.
Well, you can get some information, and as long as you treat it as just basic curiosity, and maybe it means something and maybe it doesn't, it certainly gets people thinking about the genetic code.
There are some diseases that we can locate, and I mean, the breast cancer gene, for example.
Well, in fact, that's the best worst example.
That's the best worst. Explain.
So a few years ago, when a gene was associated with breast cancer, it unfortunately got called in the press the breast cancer gene. We now know of at least 6 different genes that have increased risk if there's changes in those for breast cancer. By the time we finish analyzing the genome, that number will probably be 50. And there's all kinds of environmental traits that come into this. So the chance of getting breast cancer based on what anybody inherited from their parents, that explains maybe 3 to 5% of cancer. And that was unfortunate. It's the example I use how partial information is dangerous. Some women were told that based on changes in 1 or 2 genes, they had a greater than a 90% chance of getting breast cancer. And it simply was not true. Many of them would go out and have double mastectomies, trying to take control of their own lives, which is understandable. But had they had more complete information, they may or may not have done that. So we have to be very careful about partial information.
Do you think there will be a point, and when would this be, if so, when we will be able to make fairly good predictions? I realize that there's also other factors, but where genes will be able to tell us concrete things that we can act on medically.
Well, the concrete is, I think, going to be the sticky point. What our genome will give us is probabilities, not yes-no answers. So we can try and prevent disease in the future by understanding what we're at risk for, but it's only a risk factor. It's not yes or no answers.
Well, you do mention before you went ahead and decoded your own genome that you were concerned about insurance companies, and you, after asking around, or I guess thinking about it, you realized that in fact it wasn't going to be a factor, but it could be a factor. The kind of misinformation or misreading that the public has, I assume, corporations are likely to bring to bear on people.
Well, it's certainly a concern, and there's been a bill pending in Congress for almost 10 years now. It's passed the Senate. It has a couple hundred co-sponsors in the House, but a group of Republicans won't let it out of committee for a vote. It's the genetic non-discrimination bill. Yeah. It would actually block employers from using your genetic information as a criteria for either not employing you or employing you. As more and more companies go to self-insurance for health insurance, the worry would be if they believe this data on breast cancer and that what this misinformation was, that they might hire somebody that doesn't appear to have that risk because it would save them money in the long run. In fact, it won't turn out that way. When we get our genome sequence, we'll all find we're at risk for something, which will make us probably more of an equal risk for insurance.
That's an interesting point. I mean, I was struck reading your book, and I guess I will mention it now. A Life Decoded, My Genome, My Life. Very interesting book in many ways, both from the point of view of the science and also the personal side. In it, you talk about various aspects of your own genomic profile that are make you at risk for certain diseases and so forth. And I wondered how you felt before you went ahead, whether, I mean, after all, now you're pretty informed and you understand, but at the time, there must have been a certain trepidation, I guess I feel, a sense of predetermination if you can read your genome and know what lies in your future and why you are a certain way too, as opposed to having shaped yourself. Did that come into play?
No, in fact, it was quite the opposite, because those were the concerns that many people had in fear about if we sequence somebody's genome, it might be disclosing far more about them than they would want to have disclosed. But that was, as you say, a deterministic point of view, and that came out of some of the early thinking in genetics. We know we're not a genetically deterministic species. We're much more than just what our genes indicate our possibilities are in life. Knowing that, I was not afraid to have my genome done, and that's one of the reasons I chose to do it, to try and set an example and put it on the internet. It's easier to do that as an individual than asking you to do that. And so I tried to do that with leadership, but also just amazing scientific curiosity. What's there, and can I understand more about myself from looking at it?
Well, it struck me that it was very reflective of your personality, your whole style of conducting research and so forth, that you would use yourself in this. And I wonder what kind of flack you got or response you got from your peers on that score?
Well, there's a long history in science of scientists doing the risky experiments on themselves. So there's been varying responses to people recognizing it for what it was, to claims that I have the largest ego on the planet because I sequenced my own genome. So it's
so you had various positions.
So there's a range of positions.
Well, this book is so interesting because, along with charting a scientific breakthrough, it also is a story of weaving your way through the scientific establishment, as well as through the corporate establishment, the private and the public. And I would say, and perhaps that you've had problems equally with both sides of that equation. Could you tell us a little bit about the politics of doing what you did and how much stress that involved?
Well, let me preface that by, and I was talking to a student group just earlier today, that I tell all students if they're not using their intellectual talents to come up against and challenge dogma and challenge pre-existing assumptions, they're not doing their job. And I ran up against both of those in terms of trying to change a very large scientific establishment that was set with a multi-billion-dollar program and wanted to just proceed with a large public works project.